Imagine a world where the secrets of plant growth are unlocked with the help of cutting-edge technology. This is not just a dream, it's becoming a reality through the innovative use of UAV, unmanned aerial vehicle phenotyping, which is shedding light on the genetic factors that shape the architecture of cotton plants. But here's where it gets controversial. While traditional methods of measuring plant traits are slow and labor-intensive, this new approach promises to revolutionize how we understand and enhance crop yields. Cotton, scientifically known as Gossypium spp, is a vital fiber crop that plays a significant role in the global textile industry. The success of cotton farming heavily depends on specific plant architecture traits, including height, branching patterns, and stem growth, all of which directly influence yield and harvesting efficiency. Upland cotton, G. hirsutum, which constitutes more than 90% of the world's cotton production, particularly benefits from optimized plant height. This optimization allows for denser planting and facilitates smoother mechanical harvesting. Historical agricultural advancements, such as the incorporation of dwarf genes into staple crops, have demonstrated the substantial benefits of modifying height-related traits. However, the traditional breeding process for adjusting plant height is often slow and requires extensive fieldwork. With the rapid progress in genomics, pinpointing the genes responsible for cotton height has emerged as a critical area of research. Yet a significant hurdle persists: the need for accurate, high-throughput field data on plant height that can keep pace with the vast genetic datasets being generated. A recent study published in Plant Phenomics on March 5, 2025, by a team led by Zhuren Yang from the Chinese Academy of Agricultural Sciences showcases how UAV-based multisensor platforms can effectively address the challenges of field phenotyping. In this groundbreaking research, the team utilized UAVs equipped with RGB cameras and LiDAR sensors to analyze 419 cotton accessions at 5 different developmental stages. They extracted plant height pH values using linear regression and machine learning techniques, subsequently validating these results against manual measurements. The findings revealed that second-degree polynomial regression provided the best fit, achieving impressive R-squared values of 0.8974 for RGB-derived plant height and 0.9173 for LiDAR-derived measurements when compared to traditional ruler-based assessments, all while maintaining low error rates. Even when tested on all 419 accessions at later growth stages, the correlation between UAV predictions and manual data remained strong, particularly for LiDAR measurements, which showed R values of 0.89 and 0.87 at 2 different time points. To enhance prediction accuracy, the researchers evaluated 12 machine learning models, finding that least angle regression for RGB data and Huber regression for LiDAR data yielded the best results, with R-squared values of 0.914 and 0.934 respectively, demonstrating a robust correlation with field measurements. Building on these reliable estimates, the team employed k-means clustering to analyze the dynamics of plant height, categorizing the cotton population into 5 distinct growth patterns, each characterized by unique trajectories of height increase and peak timing. Following this, a genome-wide association study, GWAS, using UAV-derived plant height predictions, identified 1,465 single nucleotide polymorphisms, SNPs, linked to plant height. Notably, 5 consistent genomic signals were found across chromosomes A01, A02, A07, A10, and D11. Within these regions, 34 annotated genes were identified, including GHPH1 and GHUBP15, both of which are already recognized as regulators of cotton height. This discovery not only validates the effectiveness of the UAV phenotyping platform, but also led to the identification of a novel gene, GHPH-UAV1, which exhibited significantly reduced expression in a dwarf mutant compared to wild-type cotton. Functional validation revealed that overexpressing GHPH-UAV1 resulted in a 16.48% increase in plant height, while knocking it out using CRISPR-Cas9 led to a 21.44% reduction in height. along with corresponding changes in stem cell length. These findings collectively illustrate that UAV-based phenotyping, combined with sophisticated data analysis, serves as a powerful and scalable tool for unraveling the genetic regulation of complex agronomic traits. By generating rapid, cost-effective, and precise time series data, researchers can forge stronger connections between genetic variation and crop performance in real-world conditions. For cotton breeding, the identification of GH-PH UAV1 presents a promising new genetic target for refining plant architecture, ultimately aiming for higher yields, improved resistance to lodging, and better compatibility with dense planting and mechanical harvesting. This innovative strategy holds potential not just for cotton, but could also be applied to other essential crops like maize, wheat, and rice, where plant height remains a fundamental trait influencing agricultural productivity. What do you think about the implications of UAV technology in agriculture? Could this be the future of crop breeding, or do you see potential drawbacks? Share your thoughts in the comments.